So our first speaker today is going to be Jimmie Ye. Jimmie has been a faculty member in the Department of Medicine at UCSF since 2015. His background is in bioengineering and computer science. He did his postdoctoral work with Aviv Regev at the Broad. In addition to the Genomic Immunology Institute, he is a member of the Institute for Human Genetics, the Bakar Computational Health Sciences Institute, the Parker Institute for Cancer Immunotherapy, and the Chan Zuckerberg Biohub. His research focuses on harnessing the power of single cell and computational biology to study and model how human immune cells sense and respond to their environment. And his talk title today seems to have changed to multiplexed multimodal single cell immunology. So go ahead, Jimmie.
Right, great. Thank you so much, Stacy, for the introduction. I just want to say that I'm very excited to be, I guess, somewhat of a senior member of this new institute. And what I'm going to do today is share with you some of the work that my lab's been doing for the past 5 years or so, building new single-cell genomic technologies and then applying them to study increasingly more complex immune-mediated diseases. And so, I like to start with this slide that I actually show a lot of my first-year graduate students to just, get them up to speed with what's going on in the field. Single-cell genomics certainly has taken over in terms of being the de facto method for querying complex biology in the last decade or so. What this plot shows, adapted from a 2018 paper, is on the x-axis time, on the y-axis the log number of cells that's been sequenced by the community. And what this indicates is basically exponential growth in the number of cells that we are able to profile per experiment. And it's reminiscent of what we could do with genome sequencing about a decade ago. And so whenever you see sort of exponential growth like this, it's almost always driven by technological advances. And so our sort of story will start with massively parallel single-cell sequencing. And this is a particular approach where we use DNA barcodes to label single cells in physical compartments, popularized by a local Bay Area company called 10x Genomics, which encapsulates cells into these microfluidic droplets. And so, you actually hear quite a few talks later on that leverages 10x Genomics and single-cell sequencing to study the immune system in autoimmunity as well as cancer. And I just want to highlight a paper that we worked on last year with Larry Fong, which I think highlights some of the advantages of being at UCSF and being able to take some of these new technologies and apply it to study unique patient cohorts who are being treated with some of the newest drugs on the market. And so in this case, we're taking a cohort of bladder cancer patients who are actively being treated with anti-PD-L1, so checkpoint blockade. And one of the first things we looked at was just asked, what are the frequencies of CD4 positive helper T cells and CD8 positive cytotoxic T cells in the tumor? And what we were surprised to find, just with standard technology, flow cytometry, was that CD4 T cells were more abundant. And we used then single-cell sequencing to query in an unbiased way the states of CD4 T cells in bladder cancer. And we identified a few different types of CD4 T cells that are expected, including this orange cluster and this green cluster, which are regulatory T cells that are distinguished by their expression of IL-2RA or CD25. But what we were really surprised to find were these two populations of cytotoxic CD4. So these are CD4 T cells that basically behave like CD8 T cells, at least according to their transcriptome, and they're distinguished by the expression of different granzymes. And so cytotoxic CD4s have been previously described in vitro, a lot of them, and in chronic infections. There's some thought that they're just an artifact of culture. So we were curious as to whether or not these cells could actually function as killer T cells and neutralize tumor. And so what we did is we basically took tumor cells and grow them in culture, and they will grow, and there's some low level of death. And then we will co-culture these cells with CD4 T cells at a ratio of 30:1, and you can see that the death increases. Now, remember, CD4s contain both regulatory T cells that are immunosuppressive as well as these cytotoxic T cells. And if we only grow the tumor with cytotoxic T cells, you can see that these T cells neutralize the tumor even more efficiently than all CD4s. Now, the difference between CD4s and CD8s is that CD4s recognize their cognate partners through MHC class 2. And so we were wondering if they still neutralize tumor through MHC class 2 binding or recognition with the antigen. And so when you block the CD4 T cells with anti-MHC antibodies, you can see that the killing is basically now attenuated. And so this is using standard technologies, querying admittedly a rare disease with a severe phenotype, if you want to think about it as a geneticist. But my lab actually thinks a lot about much more complex diseases, in particular autoimmunities. And so I'm going to describe some technologies that we've built to leverage single-cell genomics to query much larger population cohorts and to study complex diseases like autoimmunity. And so the particular disease that we're interested in studying is systemic lupus erythematosus, which is a systemic autoimmune disease with multi-organ manifestations. And what we expect to see for a heterogeneous disease like this is that, if you think about the distribution of cells in some low-dimensional manifold, which some of you may be familiar with, this is just projecting cells in UMAP space. Maybe we don't expect a brand new population to pop up like the cytotoxic CD4s, but that there might be subtle shifts in cell state between cases and controls. And to really capture these subtle shifts, we would need large sample sizes. Another application that my lab thinks a lot about is looking at the genetic effects of gene expression and also to contextualize those genetic effects in specific cell types. And we know that many genetic effects, especially the ones that are non-coding, would be, again, small, will lead to small changes in gene expression. And so to really query that, you would also need large sample sizes. So the question is, how do we do single-cell sequencing at scale? And so what we need to do is basically some new method innovations. And the first method I'm going to talk about is MUXI, which is actually quite easy to implement. Because what it does is it basically leverages the standard microfluidic platform that 10x Genomics has built. But instead of sequencing one sample at a time, we're just going to mix samples that are genetically different and sequence them together. It turns out that there's enough genetic differences contained in the transcriptome of a single cell that, given the genetic information for all of the samples that went into the pool, we can uniquely assign each cell correctly back to the donor of origin. And we can also identify cases where multiple cells from different individuals are simultaneously encapsulated and clean up the data this way so that we get many more bona fide single cells. So you can read about the method, but just some key points about this is that we basically, by doing this, can increase the current throughput for single-cell genomics by about 5-fold. That reduces the cost significantly and will allow us to use these new technologies to query large population cohorts. Sample information is automatically encoded, and it's extremely accurate and simple to do. All we do is take cells from different people and mix and perform single-cell sequencing and run a piece of algorithm that will do all the work for us. And it's applicable to other types of single-cell sequencing assays as long as we're querying some amount of genetic information, including single-cell TAC-seq. And importantly, for downstream analysis, our approach minimizes batch effects and just generates better data and allows us to do proper randomization. And so we applied this method to study lupus. And this is a cohort that we generated in collaboration with the rheumatology division here. It's a total of about 250 samples, 160 patients, 99 controls. We were targeting about 5,000 circulating immune cells or peripheral blood mononuclear cells per sample. The cohort is primarily of East Asian and European. And lupus affects females more than males, and so our cohort is predominantly female. In total, we generated one of the largest single-cell sequencing data sets ever collected in humans, in about 1.2 million cells. In addition to measuring gene expression for a limited set of samples, we also were able to get surface protein profiling for 100 surface markers. So here's what 1.2 million cells look like. Again, what we've now done is take a 30,000 dimensional data set, one for each gene in the genome, and reduced it down to 2 dimensions. These are arbitrary. They're labeled as UMAP1 and UMAP2. Each dot here represents a cell, and so there's 1.2 million dots. And cells that are closely related to each other in terms of their function are going to be close to each other in this arbitrary 2-dimensional projection. And what do you expect to see or we're excited to see the expected populations of myeloid cells and lymphoid cells. And now if we actually take that data and project it separately for the cases and controls, we can see what I mentioned earlier is that it's not like lupus patients have a brand new cell type, but instead they certainly have all the cell types that we expect to see in the controls, but there's a subtle shift in the distribution of various different types of cells. And this is consistent in both cases of European and East Asian ancestry. So, but we wanted to assess in an unbiased way how quantitative this data set is going to be. And so what we did is first take a step back. We're going to group all of these myeloid populations, these 4 populations, into a myeloid bucket, and then the lymphoid populations into a lymphoid bucket, and look at the frequencies of myeloid versus lymphoid cells. And as expected, we detected lymphopenia. So in lupus patients, we have fewer adaptive immune cells and more innate immune cells. Now, how do we know this is quantitatively accurate? Well, it turns out, absolute monocyte abundance and lymphocyte abundance are 2 metrics that we collect when we get a complete blood count on our patients. And this is from a separate blood draw that doesn't require any sophisticated single-cell sequencing. So we correlated our estimates of monocyte and lymphocyte abundance with what's reported in the electronic health record from months ago. And we were very encouraged to see that for monocytes, except for these 2 outliers, it's very well correlated. And for lymphocytes, it's even better correlated at an R-square of about 0.95. So single-cell sequencing is incredibly quantitative. And we, it's no trivial task. We're taking frozen blood, thawing them, multiplexing, sequencing, running an algorithm, clustering, and then doing this aggregated analysis. Now, if we just wanted to figure out that lupus patients have lymphopenia, we certainly wouldn't have to do single-cell sequencing. But the power of the method is that we can now ask what are the individual populations that's changing. And what we identify is that's really the CD4 positive T cells that are now less abundant in lupus patients compared to healthy controls. And there's a concomitant increase in the classical monocytes, but this is just because single-cell sequencing is relative in terms of proportion. So the absolute abundance of monocytes is not changing, but the frequency is increasing. If you compare cases of European versus Asian ancestry, for most of the major populations, the changes in composition are the same, except for these CD4 T cells. In Asian patients, it's even more depleted than European patients, and it is not a function of treatment. So there are actually a handful of patients who haven't been even on steroids for the past 6 months, and they also exhibit a decrease in CD4 percentage. We can take the lymphoid cells and dig even deeper and subset of various different populations of lymphocytes and further refine our understanding of changes in composition. And it turns out it's that the naive CD4 T cells that are really lost in lupus patients and other CD4 populations are not changing in frequency. So that's like composition. One of the nice things you can do with single-cell sequencing data is that we could, in addition to look for changes in composition, we could ask for changes in cell state. And so the first thing we did, and lupus, of course, has been studying for quite a while, is we just tried to recapitulate what's already known about changes in immune cell state in lupus patients. In particular, a subset of lupus patients have been known to overexpress a set of genes that are related to type 1 interferon response, usually upregulated response to a virus. So if you just take all of our data and collapse it, and now simulate what would have happened if we actually did a bulk RNA sequencing experiment, we recapitulate what's already known, which is that there are these 2 clusters of genes that actually look very similar in terms of their distribution across individuals that are upregulated in lupus patients, right? And it's only a subset of lupus patients. So red here are the patients, about 50% of patients have higher expression of these type 1 interferon stimulated genes. Now, what's really nice with single-cell sequencing data is that we can further annotate these genes into 2 clusters. The first one that we call PANUP is overexpressed in all major immune cell types, but the second blue cluster is only expressed in the myeloid-specific cell types. And this type of information is very, very difficult to obtain if we were to do standard sorting and sequencing, but with single-cell analysis, we can get this in one run. Another highlight of what we can do when we do this unbiased profiling of circulating immune cells is that we can start correlating changes in cell state with changes in composition. And so I mentioned earlier, there's a decrease in T cell abundance, naive T cell abundance, and then there's this activation of interferon stimulated genes, primarily in myeloid cells. It turns out that these 2 molecular features are inversely correlated in cases. So the more interferon activation you have in myeloid cells, the fewer naive CD4s you have in circulation. And we think this is actually consistent with the pleiotropic effects of type 1 interferon cytokines, A number of papers have described how they have a specific effect in activating monocyte antiviral response. But perhaps a slightly less appreciated result that Jason's sister and others have described here at UCSF is that type 1 interferons will also sequester CD4 T cells in secondary lymphoid tissue, which could potentially lead to a loss of these cells in circulation. So this data is all now public, and if you want to play with it or look up your favorite gene, you can access this through the Zuckerberg Human Cell Atlas portal. And so in addition to sort of autoimmunity, we've recently taken this approach to also study COVID. So here again is a set of genes that are overexpressed in a set of lupus patients that are generally upregulated in response to virus. We also queried, using the same approach, COVID patients and saw that there's heterogeneity in the expression of type 1 interferon stimulated response genes. In fact, some of the most severe patients in darker red have a deficiency in their type 1 interferon response. And this has actually been described in a number of other papers. What's nice about our work is that we could show, looking at this particular type 1 interferon score, that it's only really low, or it's certainly most depleted in these classical monocytes, particularly in these severe patients with COVID-19. In a subset of these severe patients, they happen to have autoantibodies as type 1 interferons, which occurred at a frequency of about 20% of our critical patients, and can see that there are some of the patients with the lowest expression of these type 1 interferons. What we could also do is we can now profile surface markers using antibodies that are tagged with DNA barcodes. We actually applied this approach to profile PBMCs from COVID patients, and we identify a single marker called LAIR1, which is an inhibitory molecule originally cloned in natural killer cells by Louis Lanier, and that this particular marker is elevated in patients with a deficient type 1 interferon response, but not detected actually in healthy controls, right? So we think that this could be potentially a marker for patients who may have a deficient interferon response. So that's sort of using an approach that we previously developed to query autoimmunity and also infectious disease. And for the rest of the time, I just want to give you two vignettes on some new technologies that my lab is building. The first one is an approach that we've been calling cytometry by sequencing, using a combinatorial indexing strategy. And so just to go back to this slide for a second, what we're really thinking about now is how do we continue to scale the throughput of these single cell sequencing experiments so that we can get to now per experiments millions to tens of millions of cells profiles. So this is similar to what we already do with flow cytometry. And we can actually think about doing this in a standard 10x based system. So if you run a standard single cell sequencing experiment, it turns out you're wasting a lot of the reagents because of Poisson loading of cells into droplets. What we've done previously with sample multiplexing is increase the number of cells that we can put into the system, but also tolerating a higher frequency of multiple cells simultaneously encapsulated. What we've now developed is a strategy where we want to use all of the real estate. So we want each microfluidic droplet to contain a cell. In many cases, it will contain several cells, but we want to develop a strategy that will be able to resolve all of the cells that are simultaneously encapsulated. If we can do this, in theory, we can get a 50x increase in throughput using currently available platforms. So here's how we build our version of combinatorial indexing. What we're going to do is take antibodies and tag them with DNA molecules. And then we're going to use another DNA molecule that will hybridize to these antibody handles into multiple pools. And that first pool barcode is essentially, you can think about this as a birthday analogy, that this first pool barcode will give you the month of the year that a particular cell is born on. And then we're going to pool all of the cells together and then redistribute them through single cell sequencing into droplets. And that second barcode that we put onto the cells is going to be day of the month that a cell is born on. And the combination of the first index and the second index will give you the unique birthday of a cell. So this is what's going to give us single cell sequencing data. So this is what it looks like when we actually run our approach. If you take now 200,000 cells and try to profile them in a single 10x experiment, and by the way, that's about a 20x increase in throughput. If you don't use that first barcode, or you just look at the droplet barcodes, what you get is essentially a mess. There's no information here. We know that there's a number of different innate and adaptive immune cell types, but all we see is sort of a giant hairball. And we can't even separate out CD4 and CD8 T cells from CD14 monocytes or CD19 B cells. But if you use that first barcode, we can identify all of the expected populations that we expect to see in circulation. The data is actually quite comparable to other approaches for cytometry, including CyTOF. So this is a collaboration with Matt Spitzer's lab, where we use the exact same antibodies and ran CyTOF. And if we project the data using CyTOF versus CytoSeq, we get a very similar projection. But there's a number of advantages of using DNA as a barcode rather than heavy metals or fluorophores. One of the advantages is the flexibility. So in addition to sort of tagging these antibodies to be compatible with a particular assay, we could actually change this hybridization oligo to get compatibility with a number of commercially available antibody panels, but also commercially available microfluidic platforms. And just to showcase that flexibility, the initial data I showed was tagging 28 surface markers, but here we can tag 60 surface markers, or using a commercial platform from BioLegend, we can tag up to 165 markers. And the number of cells we sequence, we also push the throughput a little bit. And now we're getting potentially hundreds of thousands of cells through a single 10x experiment. Again, the throughput here is now much, much closer to flow cytometry. The data is actually very quantitative. We use the same 10 donors to do the first experiment and the second experiment. If you look at cell composition on top, as well as intra-individual variation in specific markers for specific cell types, they're both very well correlated, suggesting that we're capturing intra-individual differences in composition and gene expression. We can also combine this with other multimodal profiling approaches. So in addition to getting surface protein abundance, we can also get RNA data out. And so now with one assay, we can get high throughput single cell genomics for both the RNA at the bottom here, as well as surface protein expression. So that's one approach that we're very excited about, applying this to allow us to do some new experiments in immunology. The second approach that we recently built is to try to spatially resolve single cells. So I think many of us can probably appreciate that in complex tissue, the distribution of cells is not random. So this is just looking at a tumor, and the infiltrating immune cells are not randomly distributed with tumor, right? So here in pink are the macrophages, and they seem to cluster. And then in yellow are the cytotoxic T cells, and some of them seem to cluster, others seem to be somewhat randomly distributed. When you do single cell sequencing, we lose all that spatial information because we have to dissociate cells. And then when we actually get the single cell transcriptomes out, we can't take each cell and localize it back to space. So we wanted to solve this problem. And this is a collaboration with Alex Morrison and Eric Chow at the Center for Advanced Technology. And the way we're going to solve it is actually a very simple idea, is that we're going to make these hexagonal wells. We've made 500 micron wells, but we can also make 200 micron wells. And we're going to dissociate tissue into these wells. Each of these wells will be barcoded with the DNA molecule. And then we're going to use the same combinatorial indexing approach I talked about earlier. So here, the spatial barcode will be that first barcode or month. And then we are going to, after labeling the cells, we'll pull them up and redistribute them into a second set of physical compartments. And that second barcode will be the day of the month. And so again, the combinatorial index of the spatial barcode and the PCR barcode will give us a unique birthday for each cell. When you run this assay, what we get out is bona fide single cell sequencing data in the bottom, but we can also resolve for each cell where in space at the resolution of the wells that we make, each cell came from. And so here's what it looks like. This is a liver tumor model where we take mouse liver and injecting MC38 tumor, and we can take a slice of that tumor and digest it onto our XYZ array. And we also can stain that tumor and show that the liver here is going to be stained red and the tumor is going to be stained yellow. When we perform XYZ, we get single cell sequencing data here on top. We see the expected populations, including MC38 in yellow. Those are the tumor cells, as well as the hepatocytes in red. So as a sanity check, we just wanted to see whether or not the hepatocytes in the tumor cells are localizing in space, similar to what we observed with the H&E stain. And encouragingly, they do. Liver cells are highly enriched in that light or in that red region, and then the tumor cells are enriched in the yellow region. Okay, so what can we do with all this information? Well, what's the benefit of space? When we have single cell sequencing data that's spatially resolved, we can start asking questions about what cell types are co-localizing in space. So in addition to these liver cells and MC38 tumor cells, we also have a number of populations of immune cells. And in particular, there's these two types of innate immune cells, the Cooper cells, as well as these myeloid cells in sort of this light green. If you look at the spatial distributions of these cells that are actually phenotypically fairly similar to each other, we can see that they're quite differently distributed. And in fact, the myeloid cells are co-localizing with the MC38 tumor, versus the Kupffer cells are co-localizing with hepatocytes. And that makes a lot of sense because the Kupffer cells are also annotated as liver-resident macrophages. And so there's also a number of spatial sequencing platforms out there. What's the benefit of actually getting bona fide single cell data? And so one of the benefits is illustrated here. So one of the features of having spatial transcriptomic data is we can ask, what are the gene modules that are differentially expressed with respect to space? So here are two sets of genes that appear to be higher expressed in the tumor region versus the liver region. So there's a tumor response module and an immune regulation module. And when we get data like this, usually it's very difficult to then pinpoint the cell types that's actually driving these signatures. But when you have bona fide single cell sequencing data, we can overlay the expression of each of these signatures onto the single cell sequencing data and see that the tumor response module is in fact expressed by a subset of the MC38 tumor cells themselves, and then the immune regulation module is expressed by the myeloid cells that are co-localizing with the tumor. So I'll summarize. My lab has been somewhat obsessed with developing technologies, new technologies that we think will allow us to interrogate the immune system in brand new ways. I showcased an approach that we developed a few years ago to enable population scale single cell genomics. We applied it to study a complex autoimmune disease called lupus. We think that this approach is an unprecedented opportunity to capture simultaneously variability in cell composition as well as cell state. We discovered that the overexpression of type 1 interferon response genes in myeloid cells in lupus patients is inversely correlated with the abundance of naive CD4s, and this is consistent with the pleiotropic effects of type 1 interferon cytokines. I didn't talk about this, but we also identified some interesting heterogeneity in granzyme H positive CD8 T cells in lupus patients. I also didn't talk about how we can use this data to now increase the resolution of gene expression mapping, identifying the genetic basis of gene expression, and annotate disease-associated variants. But I'm happy to share some of those results offline. And then I introduced two new methods that we're really excited about. CytoSeq, which will allow us to scale using sequencing the number of cells and the number of features that we can measure simultaneously from immune cells. We're currently thinking about extending CytoSeq to allow us to profile the entire exoproteome because DNA tag antibodies or nanobodies well, DNA can be used to tag as many nanobodies as we want. Some additional projects ongoing in the lab using an approach is we can now contemplate these genome scale perturb-seq experiments. So we can knock out every single gene in a human genome and profile the consequences of those perturbations on surface protein in T cells, for example. And then finally, XYZ, which is a spatially resolved single cell sequencing approach. We're extending this to try to get to multimodality, to simultaneously measure gene expression as well as surface protein expression or chromatin accessibility, obtain FFPE compatibility, and also to be able to get immune repertoire sequencing, and then aspirationally to be able to track individual cells that are genetically perturbed and see where they're going in space. The big picture sort of, or the big question that my lab is going after with some of these new methods is to try to map coordinated changes in cell composition and cell state. We think these are unique features of multicellular organisms like humans, and certainly will be a next frontier in complex cellular biology. With that, I'll just leave my acknowledgements up and happy to take some questions.